The overall goal of the following experiment is to conduct a high-throughput genome-wide screen of microRNA targets using the Target ID library. This is achieved through cell transfection with the library plasmid, a genome-wide collection of cloned cDNAs. Transfection is followed by selection for stable cell lines. The established library cells are then transfected with the microRNA expression construct to allow co-expression of the microRNA target and thymidine kinase. At this point, ganciclovir is used to select for cells expressing specific microRNA targets by selecting against thymidine kinase expression. The cDNA containing microRNA target sites are then PCR amplified using the Target ID amplification primers in order to sequence and identify the microRNA library targets. Ultimately, results are obtained that identify specific microRNA targets in cells based on deep sequencing analysis.
The Mission Target ID library is designed to assist in the discovery and identification of gene targets for microRNA. Here, we demonstrate its use and application.
Prior to the start of this protocol, choose a cell line that does not express or expresses low levels of the miRNA of interest. Culture and expand the cells to obtain approximately 2 × 10⁷ cells at 80% confluency. After trypsinizing the cells, transfer them to a